In this video, we're going to go through some revision notes for Biology 0610 Cambridge IGCSE for examinations in 2024 and 2025. So let's begin. So this topic is topic 13, which is about excretion. So let's first define what is excretion. Excretion is the removal of waste substances of metabolic reactions inside cells and toxic materials and substances in excess. So inside cells, there are waste substances of metabolic reactions, and excretion is the process of removing these waste substances inside or caused by metabolic reactions and other toxic materials and substances present in excess inside our bodies. So the three main organs that are involved in excretion in our syllabus is the liver, the lungs, and the kidneys. As you can see in the diagram here, the lungs mainly excrete carbon dioxide. The lungs excrete carbon dioxide as a waste product of aerobic respiration during exhalation. Kidneys mainly excrete excess water, salts, and urea. Kidneys excrete excess water, excess salts, and urea formed in the liver from excess amino acids by producing urine. Coming to the carbon dioxide and excretion. So carbon dioxide dissolves in water and becomes acidic. By this, it lowers the pH of the cells, and this reduces the enzyme activity, because if the enzyme moves too far away or too close to from the optimum pH, it begins to denature. And what carbon dioxide does now, carbon dioxide, when it dissolves in water, it makes your blood acidic, and it causes the enzymes to lower its pH. This will eventually end up in the enzymes being denatured and losing the shape of their active site. So too much carbon dioxide and urea in the body is toxic, and it must be removed. Thankfully, the urinary or excretory system helps us in doing this. So these are the urinary system structures, beginning with the most famous one, the kidney. The kidney is a tube-bean-shaped organ that filters blood. Next is the ureter, a tube that connects the kidney to the bladder. Bladder is the organ that stores urine, excess water, salts, and urea produced by the kidney. Urethra is a tube that connects the bladder to the exterior, where the urine is released. By exterior, we mean the penis here. So this is an illustration of the urinary structure diagram. As you can see, we have the vena cava and the aorta connected to the heart. The kidneys are here, two bean-shaped organs to filter blood, connecting into the bladder from the ureter and renal vein, going down to the urethra to be excreted. The functions of the kidneys are located in the back of our abdomen. The functions of them, the first one, they regulate water content in the blood. This helps to maintain blood pressure. They also excrete toxic waste products of metabolism like urea, salts, and etc. So waste substances that need to be removed from the blood include urea, creatine, ammonia, uric acid, excess hormones, excess water, and excess salt and ions. These could exist as sodium, chlorine, magnesium, calcium, and hydrogen carbonate. This is a detailed structure of the kidney. The kidney contains the adrenal gland on the outside. After that, jumping inside the structure of the kidney, we have the cortex, then these bean-shaped structures inside, or these triangle structures, are known as the medulla. The nephrons are located between the cortex and the medulla, and the midsection of the kidney leads out to this yellow section, which is leading out to the bladder. Blood comes in from the top and goes out from the bottom. Nephrons. So nephrons are also known as kidney or renal tubules. These are tiny structures inside your kidneys used for filtration. So we know that the kidneys filter blood, and the part of the kidney that does the job of filtration are the nephrons. Nephrons start in the kidney cortex, and they loop down into the medulla and back into the cortex. The contents of the nephron drain into the innermost part of the kidney. This helps to collect urine, and then it flows into the ureter and carried into the bladder for storage to be excreted out of the exterior. This is a detailed structure of the nephron. So the nephron, on the left side, you have the proximal convoluted tubule connected to the glomerulus in the Bowman's capsule. On the right hand side, you have the distal convoluted tubule. The distal convoluted tubule extends downward into your collecting duct, and both of them are connected by the loop of Henle. So the process of ultrafiltration in nephrons filter blood by the process of ultrafiltration. So arterioles first branch off the renal artery and lead each nephron, forming a knot of capillaries, the glomerus. So arteries are gonna branch off as you can see on the left hand side. Arteries are gonna branch off somewhere here, and they're gonna come together to form your glomerus inside a Bowman's capsule. As capillaries get narrower, they move into the glomerus, and blood pressure inside them increases. This increase of blood pressure is going to cause smaller molecules to be forced out of the capillaries into the Bowman's capsule, where they form the filtrate. Substances formed out of the capillaries include glucose, water, urea, and salts, but only the useful will be reabsorbed into the blood. So from these four substances, only the useful substances are going to be reabsorbed into the blood. The excess is going to be removed as waste. Process of ultrafiltration by nephrons using the diagram. So you have the glomerus here. This is the blood in the glomerus that forces out substances out of the blood into the Bowman's capsule. The nephron is used for blood supply for reabsorption. So salts are reabsorbed at the loop of Henle into the blood by active transport and diffusion, water at the collecting duct by osmosis, depending on how much H2O the body needs at that moment. So this is called selective reabsorption. Selective reabsorption is reabsorption of salts in the loop of Henle. The loop of Henle is that loop that connects the proximal convoluted tubule and the distal convoluted tubule. So salts are reabsorbed in the loop of Henle by active transport and diffusion. If you don't know what these are, referring back to topic 2, a brief reminder: diffusion is the movement of particles from a high to low concentration, moving down the concentration gradient without using energy, instead using Brownian motion of particles through a partially permeable membrane. Active transport is the movement of particles from a low to high concentration gradient, moving against the concentration gradient, using energy from respiration and using protein carriers to transfer molecules. On the other hand, water is, of course, absorbed by osmosis, which is the only way for water to move. Osmosis is like the diffusion of water. So again, coming back to the four substances: water, salts, glucose, and urea. Water is absorbed at the loop of Henle by osmosis. Glucose is absorbed at the proximal first convoluted tubule. Urea is not absorbed because urea is excreted, and salts are absorbed in the loop of Henle. Selective reabsorption of glucose. So we found out that selective reabsorption of salts and minerals is by diffusion and active transport. Water is by osmosis. Selective reabsorption of glucose. So after the glomerular filtrate enters the Bowman's capsule, glucose is the first substance to be reabsorbed at the first proximal convoluted tubule by active transport. Of course, glucose molecules are too big to be transported by diffusion, and they need energy, so energy from respiration and using carrier proteins transfers or reabsorbs glucose using active transport. Nephrons are adapted with many mitochondria to provide enough energy for selective reabsorption of glucose using active transport. Reabsorption of glucose cannot take place anywhere else in the nephron except the first proximal convoluted tubule. Diabetes and glucose. The first check by a doctor to check if a person is diabetic is the presence of glucose in urine. Why? Because glucose is something that should be absorbed, and the first thing to be absorbed in the first proximal convoluted tubule. If it is not absorbed, this means that there is presence of glucose in your blood. So diabetic people cannot control their blood pressure, hence not all glucose filtrate is reabsorbed into the blood from the first proximal convoluted tubule. Since there is nowhere else for glucose to go, it ends up in the urine. Excretion by deamination of amino acids. So when digested food molecules are absorbed into the blood from the small intestine and carried to the liver for assimilation meaning to be converted into molecules that the body needs, some excess amino acids are reabsorbed into the blood and cannot be stored, hence they are broken down by deamination. What is deamination? Let's have a look at the process. Deamination starts by enzymes in the liver splitting up amino acid molecules. Part of the molecule containing carbon is converted into glycogen and stored. Other part with nitrogen is converted into ammonia. But since ammonia is highly toxic, it is immediately converted into urea, which is less toxic. This urea will then dissolve into the blood and to the kidneys for excretion. and also a small amount of urea is released in sweat. So to summarize, nitrogen containing amino group is removed and converted into ammonia, then for urea for excretion. There are some toxic consequences of high urea, which is cell death, deposits inside blood vessels, and reduced response to insulin, leading to diabetes, which is high amount of glucose in your urine because it is not absorbed in the first proximal convoluted tubule. And that was it for this chapter, topic 13 in biology 0610. If you have any questions, I'd be more than happy to answer them in the comments below, and I'll see you guys in the next one.